So I guess I could start by introducing our first speaker. We now present Alina Velimuk-Hamatova. How close did I come? Who will speak to us about electric field quenching of graphene oxide photoluminescence. You're on.
Thank you. So hello, my name is Alina, and today I will be presenting electric field quenching of graphene oxide photoluminescence. So graphene has a quite substantial interior due to its unique physical properties. However, due to the lack of optical band gap, graphene cannot be used as an active emissive optical element. But graphene can be easily modified, and one of the most widely utilized derivatives of graphene is graphene oxide. Graphene oxide contains oxygen functional groups on its surface, and these groups localize regions of sp2 hybridized carbon that lead to the appearance of a band gap. And this quantum confinement effect enables graphene oxide fluorescence in the visible region that opens large applications in bioimaging. So for particular applications, we need different optoelectronic properties. How can we control fluorescence of graphene oxide? And one of the widespread methods is chemical manipulations of structure. However, in order this material could be easily introduced in device, we need to develop some physical method. For example, applying an electric field. And here is the carbon nanotubes, one of the relatives of graphene oxide. And it was shown that due to the excitonic nature of fluorescence of carbon nanotubes, the emission of carbon nanotubes reversible decreases while applying the electric field. And here we will show that applying electric field to graphene oxide that also has excitonic nature of fluorescence can lead to reversible changes in its fluorescence. So to do this, we made two samples, a bulk sample and single flake graphene oxide sample. For bulk sample, graphene oxide was embedded into polyvinyl pyrrolidone or PVP matrix. And then this gel-like mixture was dropped and pressed between two indium tin oxide coated transparent electrodes. And then the sample was subject to electric field and fluorescence was measured. For single flake, graphene oxide aqueous solution was drop cast onto gold interdigitated electrodes, and then the sample was imaged in fluorescence microscope. So for bulk sample, here the emission spectra of graphene oxide in GeoPVP field. And then this GeoPVP field was subject to electric field and fluorescence was monitored in situ. And we can see that fluorescence intensity gradually decreases by up to 6% at the maximal field strength. And moreover, it shows the restoration after removing the electric field, even above the initial intensity. So here is representative behavior of graphene oxide single flakes. And we can see that all flakes show substantial quenching while applying the electric field. However, some flakes doesn't show the restoration after removing the electric field. But the majority of flakes, like flakes number 1, 2, 5, and 6, show the restorations, which means that we can control fluorescence of graphene oxide even on the level of single flake. So here we also develop a theoretical model of graphene oxide quenching. We consider that graphene oxide has excitonic nature of fluorescence, and graphene oxide quenching can be assessed based on the mechanism of physical separation of exciton on the surface of graphene oxide in electric field. So we calculated the potential energy without and with applied electric field. And we can see that electric field bends the potential. And it leads to increasing probability that electron can tunnel out of the well and fluorescence will not occur. So we use the Wenzel, Kramer, and Brillouin approximation and calculated the transmission probability, which is around 5.2% for the bulk geo sample. And it was pretty close to the experimental value. So this way, we can control the intensity of graphene oxide fluorescence emission on the level of the bulk sample as well as the single flake. And upon field removal, the fluorescence intensity of the majority of graphene oxide flakes was fully or partially restored. And based on the exciton model, we calculated electron transmission probability that was pretty close to the experimental value. And utilizing the electric field as a mechanism to fine-tune optoelectronic properties of graphene oxide, offers a degree of reversibility in device geometry. Thank you so much.
Thank you, Alina. We have a couple of minutes for questions. I forgot to mention that if you had any questions as we went along, that you should put them into the chat. I didn't get any, obviously, because I didn't tell you about that. But does anyone have a question for Alina? What was the most critical thing in your experiment as you were going along?
The most critical thing?
Yes.
So I would say that we didn't get like the best result. We got only 6% of quenching. And if you want, for example, apply this material for some device that I would expect that we want to get the best, the best quench, better quenching that only 6%. So it was one of the limitation for applying directly to device.
Okay, thank you.
Thank you.
As a follow-up on, why don't you ask it, Kayla?
I'm sorry?
Go ahead and ask the question. Dr. Green. I think you had a question. No? I thought I saw those in the chat, but that's okay. Our next presenter will present at 2:23.
Thank you.
Dean Williams, you have a question? I guess it's hard to ask a question when you're muted. But we only have a few seconds before our next speaker. So we now present Cameron Glore, who will talk to us about synthesis of isoleucine-based macrocycles. Cameron, you're on.
Okay, hello, my name is Cameron Glore, and today I was talking to you about the synthesis of isoleucine-based macrocycles. The easiest way to think about macrocycles are big rings that consist of 12 or more atoms. And what we know is that we can take half of that ring, which I'll call a crescent shape, add acid, and two of those crescent shapes will come together. What our research group is looking at is if we change the backbone of that crescent shape, if that affects its ability to cyclize. So this is the big idea. This is the macrocycle that I'm making. And right here, if you can see my cursor, is the amino acid isoleucine, which is my change in the macrocycle. Our goal is to reduce the side products, if possible. Also to perform as much as we can in solution. So that means kind of having one flask with a liquid and adding things in for each step, which makes the synthesis a lot easier. And if we're able to reduce side products and perform it in solution, then that cuts down on the need for purification. So this is a very simplified version of what it looks like. We have a blue protecting group a black backbone, and then a reactive acetal, which is red. This shape is normally more in a crescent shape, but I have it straightened out for simplicity. When we add the acid, these two molecules will join together and form our big ring. This is what it looks like, a little bit more complicated, showing all of the molecules. So we start with the cyanuric chloride, and then we start adding things to this. And this is what is changing our backbone. So we'll add a Bach hydrazine group, which is right here. I will add my amino acid, which is isoleucine, and then a protecting group, which is morpholane. We have gotten to this step fairly easily. And this semester I have focused on the amidation reaction, which is adding the amino acetal or different things as we find fit. So we will add an amide, which will change the amino acid to this acetal shown here. And that will be our final crescent shape that then can be cyclized. This semester, I have successfully done the amidation reaction, but this step has a lot of side products that are hard to remove through chromatography. We use column chromatography, which is our main method of purification, and these are very time consuming. So we want to find, if there are side products, we want side products that are easy to remove through extraction or other methods of purification that aren't as time consuming or difficult. So in the future, we're still working on reducing side products, and we're changing, right now I'm working on changing that last step. So instead of adding DCC, which creates a very hard side product to remove through chromatography, the graduate student in our lab and I am working on an EDC method, which is the same molecule, a little bit different, way easier side product to remove. And with this, we're always trying to increase purity and efficiency so that if this process is easy and simple, it's used more often in the industrial side, and people are less scared to try it and make these macrocycles because they can be very important and very useful in drug design. Thanks.
Thank you. I guess I'm having a problem unmuting people from here, so it's difficult. And I understand that you can't ask a question without being unmuted. So looking to see a question here, how do you determine what you have, that you have a bigger ring? Spectral analysis or biochemical properties?
So we'll do a primary testing using thin layer chromatography, TLCs, where we can see if we think that the reaction is finished or there's still reactants in our solution. And if the TLC looks good, we will run an NMR and do a full characterization on the product.
Have another, can you speak more to drug design and the difference between small molecules and macrocycles vis-a-vis disrupting protein interactions instead of blocking active sites?
So I'm not super well versed on the biological side of this, but I do know that the reason macrocycles are interesting for drug design is because they do work through a different mechanism, but the reason they're not used as often is because of their difficult synthesis. So I think kind of step one is finding an efficient synthesis. And then I know someone else in our lab group is then working on their ability for actually being used as a drug. So we're focusing more on seeing if this is even possible so that then its application can be studied more in depth.
And what sort of scale do you carry out these reactions?
So we'll start small. Right now, I am working on a 1 millimole scale, and then things are normally added in at 1 molar equivalents. And once that proves effective, we'll then increase the scale by 4 or 10 or whatever we find fit and make sure that it still works as we start increasing the amount of things going into our flask.
Okay, great. I think we'll let you off the hook. Nice presentation.
Thank you.
Kind of marking time a little bit here. Our next presentation should start at 2:31. And our presenter is Dustin Johnson. And the topic is correlation between morphology and electronic structure in as-grown microcrystalline zinc oxide specimens utilized in antibacterial assays.
Did you want me to go ahead and get started, or do you want to hold
You're
off?
on.
Okay, cool. So thank you, appreciate the intro. As you mentioned, my name is Dustin Johnson, graduate student here in the Optical Spectroscopy Lab in the Physics and Astronomy Department. So today we're going to be speaking a little bit about the correlation between morphology and the electronic structure in as-grown microcrystalline zinc oxide specimens that we utilize in antibacterial assays. So let's begin by speaking a little bit about zinc oxide, right? So zinc oxide is a very widely accessible semiconductor with many useful properties at the nano and micro scale. In particular, its wide band gap and its biocompatibility. So depicted here is the crystal structure, the anisotropic work site lattice in zinc oxide. As you can see, it has this hexagonal structure, and this structure results in two distinctive surface types. We have these polar hexagonal faces and these non-polar rectangular faces. It's also worth noting that zinc oxide is very inexpensive to produce, and its constituent elements are of great abundance. So these properties have resulted in high usage across a variety of industries and applications, from sunscreens to cosmetics, optoelectronics, and many more. At present, one of the more interesting applications is its use as an antibacterial agent. So as it stands, zinc oxide sees widespread use as an antibacterial agent. The issue is that while this behavior is well documented, it is not well understood. So many of the leading explanations revolve around surface-to-surface interactions between the bacterial cell wall and the semiconductor surface. And these are influenced by lattice properties and surface chemistry. And if this is the case, then understanding the relationship between antibacterial action, morphology, and surface charge dynamics is key to understanding kind of the fundamental mechanisms behind this observed behavior. So today we're just going to focus on one aspect of this relationship being polarity and electronic structure. So in order to do these investigations, we need a platform on which to do so. And to this end, we developed and employ a hydrothermal growth method for producing high quality crystals of tunable size, morphology, and polarity abundance. So in order to avoid internalization and better isolate only those interactions attributable to surface interactions, we focus on producing particles at the micro scale. So we were able to produce a wide range of crystals, from predominantly non-polar rod-like crystals, predominantly polar plates, and even kind of balanced prism morphology, so the whole range. By modifying the morphology of our crystals, we changed the underlying electronic structure, and it's this structure which dictates what surface interactions are available at the cell semiconductor interface. So its determination is of high interest. Specific to our application is the determination of the nature and density of the defect states present amongst these morphologies. So SEM images confirm that interactions with bacteria are in fact taking place, and it is possible that these surface defect states may play an influential role in these interactions. Our interest in these states originates primarily in the micro scale of our material. As we reduce the dimensionality of our material to 2, surface interactions dominate, and as a result, surface and subsurface defect states become highly influential. That makes understanding them an important step towards kind of fleshing out our understanding of what drives this antibacterial behavior. And to begin to address this, we must first consider kind of the cumulative effects of differing relative abundances of these surfaces on the crystal's electronic structure. And so one way in which we may do this is through photoluminescence studies. Now, photoluminescence is a contactless, non-destructive method of probing the vibrational electronic properties of semiconductor materials, wherein light is directed towards the surface, which excites electrons to higher energy levels, which is followed by subsequent recombinations accompanied by photon emissions. And so by analyzing the resulting spectrum, we pull a great deal of information about the electronic structure of our materials. So in doing so, we kind of observe some differences between the electronic structure of predominantly polar and non-polar morphologies. By comparing the spectra of two such samples at 200K, we can clearly see that the non-polar sample exhibits enhanced luminescence in both the near band edge region and in this defect region seen here centered around 2.2 eV. This increased activity in the defect region allows us to draw a correlation between defect density and morphology, which is significant as it allows us to relate morphology to fundamental electronic properties of the crystal and has impacts on our overarching goal to understand this antibacterial action. That's not the only difference we observe. If we normalize to the near band emission peak, we see a very clear 30 millielectron volt in this region's luminescence when you compare the polar and the non-polar samples. This could be indicative of a variety of structural differences, and insight into the nature of the shift will require further investigation. So in our quest to describe the fundamental mechanisms behind the observed antibacterial action of microcrystalline zinc oxide, we successfully produce microscale zinc oxide with controllable abundance of surface polarity to serve as a platform. And we employ photoluminescence measurements as a means to relate crystal structure and the electronic structure. And these studies shown that there is a relationship between not only the density of the defect states, but also other more general electronic properties of the material and the polarity. And so we will continue to our investigations and trying to characterize these differences. And we also supplement them with studies on how kind of the scale morphology changed the surface charge dynamics of our materials. And we will also run antibacterial assays. So with a clear understanding of the correlation between morphology and electronic structure, we can run a more focused investigations into specific interactions, particularly like the influence of reactive oxygen species and zinc ions, which are two leading candidates for kind of explaining this behavior that we see. And, thank you. I believe you are muted.
I don't think we can hear you if you're talking.
Be good if I unmuted myself.
Hey, you're quite all right. We got there.
Technology is terrific, and you just keep doing all these things. But I believe that we're almost at the end anyway. I was asking, what size samples did you use for your studies?
All right, you're talking about the crystals themselves.
Yes.
So they were
Did you
all.
weigh out Did you weigh them out?
We didn't weigh them. We measure kind of the size, so like the dimensions, more than the weight. But they're all micro scale. They do vary a little bit. It's not perfectly uniform, but we do make sure that they are at least larger than 1 micron, all of them.
Okay. Well, thank you very much. Our next speaker is Emery Green. And Emery Green's topic is, does emotional intelligence matter when using expressive writing interventions in a large enrollment STEM course?
Hi, everyone. I'm going to share my screen. Can everyone see that? We're
Yes.
good? Awesome. So good afternoon, everyone. My name is Emery Green, and I'm a senior geology student here at TCU. My research for this year, I hope to answer the question, does emotional intelligence matter when using expressive writing interventions in a large enrollment STEM course? So it's well known that anxiety related to academics, especially with regards to testing, is a rapidly growing mental health issue impacting students all over the world. And especially undergraduates here at TCU. These high levels of test anxiety have been shown to inhibit memory recall, reduce exam scores, and promote poor study habits. One form of test anxiety intervention, expressive writing, consists of a type of free response developed to allow students to essentially, quote unquote, dump their thoughts and emotions in written form in an attempt to mitigate these anxious feelings and other stresses they may have going on in their lives. Expressive writing has been shown to reduce this test anxiety and improve exam performance. However, the effectiveness of expressive writing may be mitigated by a student's level of emotional intelligence. This is what I explore in my research. Emotional intelligence is defined as the capacity to be aware of, control, and express one's emotions. So in order to better understand this connection between emotional intelligence and expressive writing, I initiated this research. So initially, to gain insight into the students' responses to the expressive writing prompt, I collected and coded expressive writing samples from the Geology 10113 introductory class students during the fall of 2020. I asked the students to complete a trait emotional intelligence questionnaire, the short form version of it, before the fall of 2020 semester to assign a numerical value to their approximate levels of emotional intelligence. Next, I stratified these students by quartiles into high, medium, and low emotionally intelligence groups. And then I asked these students to participate in focus group interviews, and I selected the volunteers based on their survey score and their participation in the expressive writing exercise itself. Finally, using the survey scores, I grouped the volunteers into the high, medium, and low emotionally intelligent focus groups. So if we look on the left side of my poster here, you've got purple, teal, and orange. These are representing the three quartiles of the groups that I assigned. I have 3 exemplar quotes from the focus group interviews that I had, asking them about their experiences with the expressive writing exercise. High emotionally intelligent students, for them, they definitely felt like it was a great resource to, yes, get a better measurement of how they're doing, be more aware of how they're doing going forward. Average students, nice way to vent, share anything, just know the teacher was writing something, just definitely writing down helped them. The low emotionally intelligent students, however, the gist that I got from them was that it didn't really impact them. They just did it to get extra credit for the grade purposes, as we did assign 1 point extra credit for doing the extra emotional intelligence work itself. So after completing these 3 focus group interviews, these data showed that the student responses varied substantially by group. The stacked bar graph on the right displays this distribution of codes and also the percentage of student responses by emotional intelligence group. And overall, the high emotionally intelligent students engaged with the exercise in an authentic manner, while the low emotionally intelligent students did not and generally had more cursory responses. So in the stacked bar percentage graph I have on the right, it's quite obvious that some of the more positive codes that I created, the science norms, students affected, especially value as a student, are dominated by the highly emotional intelligent students versus increased anxiety, doing it for only the extra point. Those are dominated by the low emotional intelligence group of students. And I also have some exemplar quotes noted here for a few of the codes that I created. After conducting the focus group interviews, qualitative analysis showed that highly emotionally intelligent students had more positive, authentic experiences with the expressive writing, while the lower emotionally intelligent students tended to have neutral or negative experiences with the activity. Overall, these data suggest that the level of emotional intelligence relates to how willing students were to convey their thoughts and emotions during the activity itself, which helped the higher emotionally intelligent students to have their positive experiences with the exercise. The implications of my study are that while the efficacy of expressive writing exercise is assumed, emotional intelligence is a confounding variable. Students must engage with the expressive writing in some sort of authentic manner if they are to benefit from the exercise. Thank you for listening, and let me know if you have any questions.
Again, I forgot to tell people to put questions in the chat, but I have a couple here. It says, what does authentic mean?
Authentic means the students were wanting to portray their real thoughts and their real emotions into the exercise rather than dumping something that wasn't their true thoughts and emotions in doing so. So some of the low emotionally intelligent students told me they just wrote down the word stressed or something like that, where it wasn't actually portraying their real thoughts and feelings during the exercise. So the high students actually did say specific examples of, I'm going on a stressful trip this weekend, or I have 3 tests this weekend, being authentic in the fact that they're displaying real things that are happening, real events that are impacting their lives in some way, which helped the exercise to be
I
worthwhile.
have another question. So did you have any control activity, maybe being in the higher emotional intelligence category? Just
Any
gives them
control?
a better attitude about everything. Not
Most of this, actually all of the students in the high emotional intelligence group, they all had very positive experiences. I didn't necessarily have a control group within that, but everyone that I talked to in that group all really enjoyed the exercise. Pretty much exactly what I thought was going to happen happened.
well, that's good.
Yes.
When you want things to happen and they happen, that's terrific.
That is a good thing. Yes.
All right. Well, thank you very much, Emery.
Awesome. Thank you so much.
Okay. So let's get ready for our next speaker, who's Andrew Milkosny, whose topic is evaluating sex-specific differences in cellular immune function in a small fish model, the fathead minnow. And again, I should add that please, if you have questions, put them in the chat and I'll try to use some of them. Go ahead, Andrew.
Okay, can you hear me?
Yes.
Okay, perfect. Share that. Can you see, did that work?
Good.
Okay, cool. All right, good afternoon. So my name is Andrew Milkosny and I work in Dr. Jeffrey's lab. And like you briefly mentioned, my project focuses on immune system differences between male and female fathead minnows. So brief overview of what our lab does. So we are an environmental toxicology lab. So basically we are looking at contaminants and pollutants in the environment and studying their effect on the organism and extrapolating that to the ecosystem level. So we take our fathead minnows, which is a very common model organism in environmental toxicology, and we expose them to hormones and chemicals and pathogens. And then we look at different parameters from molecular gene expression to cellular and whole organism levels to determine what exactly those contaminants or pollutants are doing. So why do we care about sex-specific differences? Why does it matter? In the study published a few years ago, they showed that almost 80% of studies do not report the sex of the fish they're using. So basically they're just saying we used a ratio of male and females, or we just used some number of fish, which doesn't tell us much. And this wouldn't be a problem if we didn't see this sort of effect. So this was a study done by another member of our lab, and she exposed male and female fish to pathogen. And as you can see here, the males are having a drastically reduced survival rate compared to females. So these studies that aren't looking at males and females specifically, they're not taking that into account. There's these huge survival rate differences between males and females that are maybe should be accounted for at baseline. And we want to know what is going on here and why is that happening? Why are males being reduced relative to females, so that the literature should be able to take that into account in the future if we know what that is. So that's our goal. We have this decrease in survival in males relative to females. We want to know what's going on. And because it's their exposure to pathogens, that tells us it's probably something going wrong or going different with the immune system. And a key component of the immune system is this thing called phagocytosis, which is the ability of immune cells to engulf and destroy pathogens. It's sort of this initial, the start of an immune response, the same thing that happens in humans. It's conserved across many different species. So we wanted to compare male phagocytic ability to female phagocytic ability. So we did this I isolated kidney cells from male and female fish, and then I incubated them with this fluorescent E. coli, and then I allowed the cells to engulf the E. coli. And then because they're fluorescent E. coli, we can measure their fluorescence, and we only want to measure the E. coli within the cell, so we added this dye that eliminates the fluorescence of the E. coli outside the cell. And if the experiment works correctly, we should see that as time goes on, we should see an increase in fluorescence as more and more E. coli get engulfed by the cell over time. And we thought that males would be having a reduced ability to phagocytose, which would lead them to not be eliminating pathogens as quickly, which would then result in them having a harmful and the pathogen damaging them and leading to their reduced mortality, or sorry, their increased mortality, their reduced survival. All right, so this is my graph. As you can see here, fluorescence is definitely increasing over time, which is telling us that the assay is working. However, there does not appear to be a difference between the just pure ability of male and female cells to engulf and destroy pathogen. On the one hand, this is somewhat frustrating as we still do not know what's accounting for that reduction in survival rate in males, but this does tell us what is not happening. And obviously we need further research to further confirm that this is not happening, but it tells us we can look at other indicators of immune function and overall function. One of those indicators being differences in gene expression. There are these things called cytokines or complement proteins. These are just core proteins involved in initiating an immune response. And an overexpression of these proteins could lead to an overactive immune response, which could then damage the organism and lead to that reduced survival that we see. This has been somewhat proposed and shown to have evidence across the literature. So this might be another possible avenue of research that our lab can take. But as of now, we do not know exactly what is causing the difference in immune function between males and females, but we are one step closer to figuring that out. Thank you. If there's any questions, feel free to ask. I can't hear.
Again, I forgot to unmute myself. Sorry.
No worries, no worries.
Question is, with the multitude of different facets of immune function that could be behind the sex difference, why did you choose phagocytosis? What led you to believe that this would be the measure to examine when, and where would you turn next?
Yes, that's a good. So phagocytosis is like, it has to happen in order for fish to have any, and same thing happens with humans, first to have any immune response. So these cells are engulfing pathogens, they destroy them, and then they take those destroyed proteins and they present them to the immune system. So this is a crucial component that has to happen in order to have all the other aspects of the immune response happen. As for further avenues of research, I think it's likely a combination of this overactive inflammatory response in males, and it's probably being caused by an overexpression of cytokine proteins, which can cause this thing called a cytokine storm, which is basically just an uncontrolled immune response that doesn't get repressed, and then that leads to multi-system organ failure. Previous research in our lab has shown that there is some difference between males and females in those proteins, but it wasn't necessarily definitive. So something further to look at.
Okay, thank you. That's great.
Thank you.
Our next speaker is Trevon Jelinek, whose topic is synthesis of organic macrocycles for antimicrobial testing. Trevon?
Yes, I'm here.
Okay.
All right. Just gonna share my screen here.
You're good.
All right. Hello, everyone. I'm Trevon Jelinek, and I will be talking on the synthesis of organic macrocycles for antimicrobial testing. So these past 2 semesters, I've been working primarily in Dr. Simonek's lab, doing the organic synthesis. Recently, I've moved into the biology side of things alongside Dr. McGilvery in her lab. And so I have kind of this joint project with these 2 mentors. And so without further ado. So most drugs on the market today are like these short molecules that are usually linear in organization. But there are some that are naturally occurring that look more like this, like a large macrocycle, as we call them. But the synthesis of these in a lab is generally pretty difficult. But we have kind of gotten a procedure in our lab to make these relatively efficiently. So most, like we use monomers in our lab. So most monomer chemistry kind of looks like this, and it's mostly polymerization with monomers just being added on to each other. But we started with a monomer base molecule like this, and then we did 3 addition chemistry reactions to add these 3 groups onto our base molecule. And this circle right here is an amino acid. And so we have been adding amino acids onto this base molecule. And so imagine that this monomer right here is depicted as this. So we make a lot of these monomers. And then the last step in our synthesis is to add acid, and we get this, our macrocycle, which is a very interesting synthesis method. And so the next question, or the beginning question actually, was what amino acid should I use to add onto here to test it on biological species? So for proteins, most of the outside of proteins are made up of these charged or polar amino acids. So to get biological interaction with these proteins in our molecule, we decided we wanted to use these molecules. So we decided to use primarily the charged amino acids. So last semester I started with glutamic acid, and that was proved to be very difficult. So this semester I switched over to using lysine. And because it's charged, we have to use a protecting group on this lysine. And so I started out the semester using this protecting group right here, which is a Z protecting group. And we had a lot of difficulty with the solubility throughout our synthesis. So we scratched that and we moved to a Bach protecting group instead. So I was able to successfully make our macrocycle using this Bach protected lysine as the addition amino acid. And now my further research Moving into the biology side of things will be to test this on cells. And so I will be testing this on this lovely bacteria. And so we're going to see if it has any cytotoxicity in these cells. So, yeah. Thank you very much to everyone that came. And thank you very much to my research professors, and as well as Jason for guiding and making some of the starting materials. So if any questions?
If there are any questions, please put them in the chat. You must have done a heck of a job. So, oh, wait a minute. Question is, do these really kill bacteria?
So I have just begun to test if they do or not. I actually, interestingly enough, did a test this week on them, and it proved to not kill the particular strain of bacteria that we're using. We're using Staphylococcus aureus, and the results were that it did not kill them. But I will do further research on this, and there's even potential to create some derivatives of my macrocycle to test those on bacteria as well. So we will see.
Yes, those were mean-looking bacteria that you had there.
Right?
Okay, thank you very much.
Thank you.
So I'm marking time here a little bit. I've got a minute left before we need to go to our next speaker. Again, we need to stay on time. So we're doing the best we can. So let me introduce the next speaker, who is Khoi Nguyen. And his topic is metrics of positive scalar curvature on Riemannian, easy for me to say, Riemannian manifolds. I hope I really kill that.
It's actually Riemannian manifold.
Good, thank you.
Yeah, his German.
Yeah,
So let me. Yeah.
bail me out.
Okay. So hi everybody. I'm Khoi and currently our junior students at TCU. So in today's talk, I will discuss you briefly about my research problem with Dr. Ken Richardson from the Department of Math on the problem of metrics of positive scalar curvature on Riemannian manifolds. So first of all, I want to talk to you briefly about the intuitive concept of curvature of curves, one-dimensional manifolds, and surfaces, two-dimensional manifolds. So in classical differential geometry, the notion of curvature is a measurement of how deviant a curve is from the usual lines, or how deviant a surface is from the usual plane. For example, looking at the curve on the left, you can see that the left portion of the curve has bigger curvature because the circle of best fit is smaller, and the right portion here has smaller curvature because the circle of best fit is bigger. Going on to surface theory, there's a concept called the Gaussian curvature, which measures the curvature of the surface. Roughly speaking, if the Gaussian curvature of a surface at a point is positive, then it locally looks like a ball. And if it's negative, then it looks like a saddle. For example, the interior of the torus viewed here has negative Gaussian curvature because locally it looks like a saddle. Now, I want to talk to you briefly about metric tensor and Riemannian geometry. So the metric tensor is probably one of the most important objects to study in Riemannian geometry, and details are shown on the slide. So basically, you can think of it as a way to generalize the Pythagorean theorem to curved surfaces. In fact, the component of a metric tensor form the coefficient of an expression used to measure length, distances, and angles on manifolds. Building off around the metric tensor, we can derive a whole host of useful formula in Riemannian geometry relating the Christoffel symbols and the two notions of curvature, the Riemann tensor and the scalar curvature, which is the generalization of the Gaussian curvature to higher dimension. Now I want to talk to you briefly about scalar curvature and volumes of balls. First of all, a geodesic is a curve on a manifold in which a particle living on it must travel in order to keep its path the straightest. For example, lines of planes and great circles on spheres. Now, a geodesic ball center at a point on the manifold is ball center at that point and half radius equal to geodesic line segment. For example, taking the north pole of the sphere as an example, one can walk away from the north pole of the sphere along great circles a distance r, and the spherical cap seen here form a geodesic ball center at the north pole of the sphere of radius r. In general, the volume of a geodesic ball will be given from the volume of that ball in R and via a formula relating the scalar curvature of the manifold seen here, which is known. In our example above, the area of the spherical cap will be different from the area of the usual circle on the Euclidean plane via also a formula relating the Gaussian curvature of the sphere. Now, I want to talk to you briefly about my research project, which is about positive scalar curvature metrics on a Riemannian manifold. So it is commonly known within the research community that on a manifold of dimension 3 or higher, we can always obtain a metric so that scalar curvature is negative. But it is not known so whether or not there is a PSC metric on certain kinds of manifold. So in this project, I will investigate product-like structures and manifolds. So I have proven two known lemmas relating to scalar curvature in different scenarios and how they interact with each other. Now I will come to my main result, the biggest result that I have proven so far relating the Cartesian products of Riemannian manifold is scalar curvature and the scalar curvature of each of its components. In the future, I will investigate manifolds where there is product-like structures of the metric at every tangent spaces and see whether or not there is a PSC metric structures on that manifold in this scenario. That's the end of my presentation. Thank you for listening. Do you have any questions or?
And if there are any questions, please put them in the chat. What made you choose this topic?
First of all, I am a geometry guy, so I love geometry. I love all things related to geometry. I think like drawing pictures is beautiful, so I want to discover more about pictures and shapes. So that's the one thing I want to study in this project with Dr. Richardson. And secondly, I want to like broaden my horizon. So I want to think like outside the box. So what else that we can do besides like normal Euclidean geometry? So I found differential geometry a very appealing track. Yeah.
I have another question. Why do products help you make the PSC metrics possible?
As I have shown here, like the product manifold, as I have shown here. So if let's say we have two manifolds and one of them is sure to have a positive scalar curvature metric, then the product, then no matter what the other one have, like it can have positive, it can have negative scalar curvature. Even if one of them has the positive scalar curvature, then the whole thing will admit appear symmetric. So the product sort of like is a very strong imposing condition on like which manifold can have like peer symmetric. As long as one have peer symmetric, then the whole thing will have peer symmetric. So
Well, there's no doubt in my mind that you're enthusiastic about this work. So congratulations. And thank you.
okay, thank you.
Our next presenter is Iman Ali. And his topic is mechanisms of zinc oxide antibacterial activity in Staphylococcus aureus. I'm not a biologist. We have just a half a minute. No, we have like 6 seconds, so I think you can get started. You can start.
Sounds good. Okay, hello and welcome to my presentation on the interaction between Staphylococcus aureus and zinc oxide. Our goal for this research was to better understand zinc oxide's antimicrobial mechanisms as it could potentially be used as an antibiotic. This is important to research with this rise in antibiotic resistance. In the US, there are more than 2.8 million cases of antibiotic-resistant infections every year, and more than 35,000 people die from these afflictions. And that's what this first graphic here is depicting. This is a major public health crisis as people around the world are dying from infections that have become untreatable. Some things that contribute to antibiotic resistance rise is antibiotic overuse, misuse, and just the general lack of research for new drugs. In 2050, it is estimated that there'll be more deaths due to antibiotic-resistant diseases than cancer. One bacteria that's notorious for gaining antibiotic resistance is Staphylococcus aureus or Staph aureus, and it's shown right here. Staph aureus is responsible for causing 90% of the skin and soft tissue infections, and these infections can become more invasive, causing pneumonia, sepsis, or meningitis. They can become fatal if the bacterium enters the lungs, heart, or bloodstream. Normally, these infections are treated with antibiotics, but this rise in antibiotic resistance makes it harder to treat. Therefore, there's an interest in the use of zinc oxide as a potential way to inhibit bacterial growth, and zinc oxide is shown right here. So previous research on the use of zinc oxide nanoparticles has shown to be effective against various bacterial species such as E. coli and Staph aureus. Additionally, it's of low toxicity to humans, therefore it makes it an even more attractive potential antibiotic. The unique thing about these particles is that they can be synthesized into different shapes, sizes, and that changes its chemical properties. So changing these features can change its activity. The issue with zinc oxide is that the mechanism of its antimicrobial activity is unknown. So we know it works, we just don't know how it works. And this is important to research because it can help us better engineer these particles so that they can be as efficient as possible at inhibiting bacterial growth. So currently there are a few proposed theories on how zinc oxide elicits its antimicrobial activity, and these are shown right here in this graphic. Our lab tested a few of these mechanisms, and today I will share one of our results with you all. So first we looked into whether particle size of zinc oxide influences bacterial inhibition. So currently the most accepted theory of zinc oxide mechanism is that the smaller particles are internalized by the bacteria and thus cause cell death. So we used large synthesized particles to see if we still got inhibition. And in order to test this, we had tubes of zinc oxide at different concentrations. So in our experimental tubes, we had equal volumes of Staph aureus and zinc oxide. And then we also had a set of tubes that had no Staph aureus just to serve as the control. We put these tubes on a rack that caused them to invert throughout the duration of the experiment. And this was just to ensure that we had constant contact between the zinc oxide particles and the bacterial cells. We then put this entire apparatus into an incubator and at 37 degrees Celsius. And then the next day we were able to obtain bacterial growth readings by measuring the optical density. So these were our results for the small nanoparticles, and this is what we expected to see as this has already been established. So we can see clear inhibition, and at 1.25 mg per mil of zinc oxide, we see absolutely no bacterial growth. And from this picture here, you can see that the particles are small enough to theoretically be internalized by the bacterial cells. We then ran this experiment with the large particles, and these particles were synthesized by John Reeks and Dr. Yuri's lab, with whom we collaborated with for this project. As you can see from the pictures, the zinc oxide particles are significantly larger than the bacteria, so there's no way that the bacterial cells could internalize these particles. You can also see that they're synthesized into different shapes, thus differing their properties. And this was our results. So for this first particle, we see nearly complete inhibition at 0.625 mg per mil of zinc oxide. Here we see that bacterial growth stabilizes around 0.313 mg per mil of zinc oxide. And then here we see more of a gradual inhibition, so we need higher doses to get more inhibition. So our results show us that these differing properties causes different zinc oxide activity. And it also suggests that internalization of the particles is not necessary to see bacterial inhibition as the current theory claims. So in the future, we'd like to test more of the mechanisms that are theorized. So maybe there's something soluble that's being released by the zinc oxide particles, such as reactive oxygen species or zinc ions that's contributing to its bacterial inhibition. We'd also like to test these particles against different bacterial types and mutants. And we'd also like to test these particles with different media types just to see how that influences zinc oxide's activity. And all of these are important in researching in order to better understand zinc oxide's mechanisms, because we could therefore then have another antibiotic that can inhibit bacterial growth. I'd like to thank the following people here for all their help and support on this project. And if there are any questions, I would be happy to answer them.
If there are questions, please put them in the chat. And it looks as though you've done a heck of a job. So no one
question.
No one seems to have a question.
All right.
So how much longer do you think you're going to be working on this?
I have a couple experiments that I'm finishing up. I graduate in May, so hopefully I'll get that done. And then one of my lab mates, Alex, is going to take over the project.
Well, that's good.
Mm-hmm.
Okay, well, thank you very much.
Thank you.
And again, I'm dragging my feet a little bit because our next speaker is due to start at 3:19. And it is 3:17:35 or so. So I believe we're staying on schedule. And while I have a minute and I keep forgetting to tell people to put questions into the chat, let me just remind everybody, if you have a question for our final two speakers, please put them in the chat for me. And I guess I got a question late, but it's okay. Let me introduce the next speaker, Holly Gould. And this topic is using GIS and remote sensing to evaluate flood risk in Houston, Texas. And we have about 25 seconds for you to mark time and get ready.
Okay. Just let me know when I can go.
Yeah, well, I can give you a countdown. Say 10, 9, 8, 7. You can start.
Okay. Hi, everybody. Thank you so much for coming to my presentation. My topic for my Applied GIS course was using GIS to evaluate flood risk in Houston, Texas. So floods are one of the most common natural disasters around the world and can cause a lot of damage to communities. So it's really crucial for communities to understand the flood vulnerabilities that they're at and develop proper flood management strategies. So the city of Houston, Texas is a major metropolitan area in the country, and they have an extensive flooding history that affects millions of people per year and really affects their infrastructure every year. So the objective of my study was to analyze these flood risks for a subwatershed in Houston, which was the White Oak Bayou watershed, and develop a flood risk map into 5 different categories. So these categories were very high, medium, low, and very low risk levels. So I did follow the methods for existing literature, and I analyzed 7 different factors that contribute to flooding. And those were elevation, slope, flow accumulation, soil hydrologic groups, average rainfall, land use classes, and then distance to rivers. So looking at my poster really quick, down here, this is all of the raw data that I collected. And this right here is the methods that I followed. So these blue boxes are the raw data that I used. Yellow boxes just signify my processing methods. And then my red box, or I guess pink boxes, those are my final products, which those final maps are shown in my results section right here. And then the last one I want to point out on my poster is this chart right here, table 2, which just provides the classification scheme for these different flood factors. And I'm about to go through how I classified each one of these flood factors. So elevation risk, which is shown in image A of my results section, so the risk was highest in the low-lying areas of the watershed. So this is because naturally water will move from areas of high elevation to low. And that is shown again in figure A. So then slope, which is shown in figure B, that slope risk was highest in the lower slope region. So again, water is naturally gonna flow off of those steeper slopes. So the lower slopes will accumulate more of that water. Next, my next factor is flow accumulation. So that is image C. And so that risk classified areas with high flow accumulation to be at higher risk. And I know that the image zoomed out you can't really tell where the risk is. It all looks green, which it essentially is, but the flow accumulation was highest along the river networks. So just really showing that the rivers had the highest flow accumulation, of course. And then next are my soil hydrologic groups shown in image D. So this risk was highest for soils that fall in hydrologic group D, which generally have slower infiltration and higher runoff compared to those in hydrologic group A. So it's more of those tightly compacted clay-like soils. And again, that is image D. So then my next one is land use. So that is image E. So land use risk is highest for the more anthropogenically influenced land types. So the developed and agricultural areas had a higher risk, and then the more vegetated natural land covers were at a lower risk. And as you can see in image E, majority of that watershed is highly developed. So it is at a very high risk. So then my next one was average annual precipitation, which is figure F, which is fairly coarse. But so this really looked at the average precipitation gradient in the watershed. So the risk classified the areas that experience more rainfall to have a higher risk. And then, compared to the areas with the lower risk levels or lower average rainfall. And then lastly, the distance to rivers, which is image G. So this risk just shows the areas that are in close proximity to river networks. And, the closer you are to those river networks, the higher flood risk you have. So with these newly classified factors, I then performed a weighted overlay analysis of all of these individual risk factors using the weights in this chart. So this is how much weight they contributed to the overall risk level. And then, so from that, I was able to generate a final risk map, which is shown in figure H, and figure I is a little bit more zoomed in. But the results do show that the majority of the watershed falls in a medium risk category with about 9.5% in high risk and then 11% at lower risk categories. And all of this is relative to the watershed. So it's the areas in the watershed that are more vulnerable to flooding compared to the others in that watershed. So looking at the zoomed in photo, you can kind of see that that a lot of the higher risk levels are more concentrated in the neighborhoods that are surrounding those stream networks. And then also the discharge location in the watershed. So this is kind of where a lot of the water will accumulate in the watershed towards that discharge area. And that makes sense because in a watershed, the natural topography is really going to drive that water to migrate to the lower lying area in a way like a funnel. And then also another area of concern, so to speak, is this discharge location in the areas of very high risk levels. Those are more centered around the downtown area of Houston. So just putting more of a higher risk at the downtown area. So that was my project. For future work. I would definitely like to do a larger project just for, a larger study area to get the whole area of Houston. I did cut it down to the sub watershed just for processing purposes. And that is my presentation, my project, and I can take any questions if you guys have any.
Very good, thank you. I'm looking in the chat to see if anyone is asking anything. And again, you must have done a marvelous job. Again, I will thank you, and we will keep moving on.
Okay,
We've
thank
got
you.
some, oh, wait a minute, time out. We've got a question. So what is the population density, neighbors / business in those high risk areas?
I actually do not know the population densities for those high risk areas. I just really was looking at, I guess, the more physical characteristics. This was a very highly developed area with a lot of residential neighborhoods. So if I had to guess, it would be a high population or residential density, but I don't know exactly.
Okay, thank you.
Mhm.
All right, well, we have a little timeout here, and I need to communicate with the tech support person. Would you please allow Deborah Rafferty to be able to share her screen for Paige Northern's presentation, because Paige is having some problems and we need to be able to show her presentation at 3:35. So until then, we will just kind of have a little break. So the next presentation will be at 3:35. And it is now 3:28. It works. So we'll be good. Are you with us, Paige?
Yes, I am.
Well, that's great. So we have your presentation up, and so let me introduce you properly and we'll go from there.
Sounds great.
Our next presenter is Paige Northern. And the title of her presentation is Lecture Fluency Impacts Instructors' Ratings of Other Instructors' Teaching. And when you're ready, go ahead.
All right, thank you. So as mentioned, my poster is about how lecture fluency impacts instructors' ratings of other instructors' teaching. So to give you a little bit of background information about this work, lecture fluency, which is just another term for the manner in which a lecture delivered, is one factor that can influence students' evaluations of other instructors. So for example, an instructor who delivers fluent lectures is typically characterized by speaking enthusiastically, walking around the room with confidence, making direct eye contact, whereas an instructor who delivers a disfluent lecture is typically characterized by showing less confidence, not very enthusiastic about content, and not making much eye contact. Now, instructors who deliver those fluent lectures typically garner higher ratings from students compared to those instructors who deliver disfluent lectures, even when the content is held constant. Now, researchers have yet to explore if instructors' evaluations of other instructors' teaching can also be impacted by lecture fluency. So it's possible that because instructors have more experience with delivering lectures, listening to high quality lectures, that they may be able to rate other instructors' teaching a little bit more accurately compared to students. However, the impacts of lecture fluency on ratings of instructors' teaching is pretty high. So it's possible that they may also be susceptible to these effects of lecture fluency. So that was the aim of the work. And to give you a bit of information about our participants, we had 44 TCU undergraduates participate in this study, as well as 42 university level instructors. These instructors were around average 45 years of age. Most of the instructors who participated were women and were Caucasian. And a lot of them had evaluated other instructors' teaching before. So for our procedure, first, we had students and instructors answer prior knowledge questions about the content in the lecture video. So the content in that video that we had them watch was about information theory, and we wanted to make sure that they didn't know a lot about that content prior to seeing this video. After they answered those prior knowledge questions, they watched either a fluent video or a disfluent lecture video. So to give you an idea about what this looked like, if you look under the materials heading on the poster, the image on the left is of the fluent lecture video, and the image on the right is of the disfluent lecture video. As you can see, the information on the screen is exactly the same, and the instructor is delivering the exact same content. So there was no difference at all in the content in the videos. The only difference is the manner in which that information was delivered. So the fluent instructor stood upright, spoke enthusiastically, and used hand gestures to emphasize points. The instructor in the disfluent video was hunched over, spoke in a monotone voice, and did not make any contact at all. So after they watched those videos, they responded to 8 evaluation items, just about how organized, prepared, and knowledgeable that instructor was. On the left, we were particularly interested in seeing if the instructors would think that that person would be a good colleague in the department, and how likely they thought they would be to receive a promotion just based upon the manner in which they delivered that lecture. So if we look at the results here, the dark green bar corresponds to those who were students and watched the fluent lecture video, while the light green are the instructors who watched that fluent lecture video. The light bluish looking bar is students who watch the disfluent video, and the grayish pinkish looking bar are the instructors who watch the disfluent video. The big picture here is that both students and instructors gave that fluent lecture, the instructor in that fluent lecture video, significantly higher ratings on all items compared to students and instructors who watched the disfluent lecture video. So overall, this means that even instructors were susceptible to these fluency effects. As well, instructors who watched that fluent lecture video said the instructor would probably be a better colleague and would be more likely to receive a promotion compared to those who watched the disfluent lecture. So overall, this means that even instructors can base their evaluations of other instructors' teaching off of this more perceptual quality of lectures instead of the actual content. So just to wrap things up, I really want to thank the College of Science and Engineering for funding this work. We received a CERC grant for it, and this work would not have been accomplished without the funds of that. So thank you very much, and I will now accept any questions.
So let me unmute myself here. Let's see here. It says, the question is, you mentioned that that an outcome in this study is effectiveness. Instructor and student evaluations typically do not explain much of the variability in learning gains. Do you have any plans to add some measure of learning? Do you have any plans to add some measure of learning into the study? Also, what are your thoughts on the medium used in this study? For example, research shows that educational videos over about 6 minutes show a rapid decline in attention and effectiveness.
Okay, so I'll answer that first question first. So yes, in other studies, we have absolutely added a measure of how much students are learning, because that's an interesting question. Are students actually learning more from this fluent instructor compared to that disfluent instructor? What we find consistently across all studies that look at this fluent versus disfluent effect is that the learning is consistent across both videos. So even though students' learning is not impacted, they're perceiving that it might be just based upon that fluency. So this definitely means that there's this mismatch there, right? Between what students think is helping their learning versus how their learning is actually occurring in classrooms. To answer the second part of that question about these videos, that's definitely a limitation of the study. It's kind of challenging to do this work in a manner other than watching videos, because then we'd have to have someone like actually setting up face-to-face and delivering these videos in the same way. But I think it's pretty relevant, especially given that we're all kind of doing this Zoom learning right now. So, I do think that there's some good points about attention dropping over time. But if anything, I think that makes these effects more impressive, because it shows at least that they're paying attention to some aspects of this lecture. I think that answers those questions, but please let me know if I didn't.
I think you did. Well, thank you, everybody, for attending. This has been an experience for me, and I'm sure for other people.